this is a big one, okay? Not just a big one in terms of length, but also in terms of the concepts that it is asking you to understand, okay? We are going to be looking a lot at what it calls partial pressures. And there will even be a couple of gas laws that are going to be kind of introduced. Basically, these are little laws that will help govern how gases move, okay? However, the main idea of this is still as simple as the stuff that we talked about in like chapter 3, which is we're going to go from an area of high concentration to an area of low concentration. That's really all we're trying to do, which means in order for us to get air into our lungs, all we have to do is decrease the concentration of the air in our thoracic cavity. We do that by making the thoracic cavity bigger, okay? Because if this is a set volume of air right now, which it is, all we have to do is make the volume larger, okay? That's it, because we have the same number of particles now in a bigger bag, which means the pressure is going to go down, okay? And when that happens, we pull air in. Then all we have to do is reverse that and make this part smaller, and it will push that air out. Okay? That's all we're trying to do. We're trying to make the chest cavity larger to pull air in, and we're trying to make the chest cavity smaller to push that air out. Okay? And as long as the air that is around your head has different oxygen and carbon dioxide concentrations than that in your lungs, which it almost always should, then we will pull oxygen into the blood and we will get CO2 out. That's all we're trying to do, okay? This chapter will make it seem like this whole process is very, very complicated. And it is, but it's also really not. When in doubt, boil it down to the simple stuff, okay? What are we trying to do? We're trying to make the chest cavity larger and smaller so that we can either pull in or push out air. And in the process, we are trying to oxygenate the blood while at the same time getting rid of CO2. That's all we're trying to do, okay? Now, we're doing this, like this is called the respiratory system because respiration is gas exchange, okay? That's what we're looking for, is this exchange of gases, particularly between the lungs and the environment, but also really between the blood and the lungs, okay? So, an exchange of gases essentially between the atmosphere and our body's cells. First off, we need oxygen. Why? So that we can do the electron transport chain. That's the main reason that we need oxygen. We need oxygen to act as an electron acceptor at the end of the electron transport chain, which is part of that whole process where we're going to go from glycolysis to citric acid cycle and then finally through electron transport chain. Oxygen comes in at that very last step to accept those electrons at the end of electron transport chain, and thus allows us to go from making 2 ATP out of glycolysis and 2 ATP out of Krebs cycle, and instead make 34. So we get something like 17 times more efficient thanks to having oxygen. Now, what just happened here? Never seen the screen before. Is it going to work? All right. So now what we need to do is we need to get that air in and we need to get that air out, which means we have to have passageways for this air to move. Okay. Importantly, these passageways have to be open all the time. This way, it is easy for the air to come in and easy for the air to go out. Okay, so we're going to have our air passageways. We're going to have a site of exchange, which means we have to have a really, really, really thin membrane in a particular couple of places so that we can have exchange happen very, very quickly. We're also going to have some other stuff happening around the same time and in the same places. We know that we breathe in through our nose, and because we're breathing in through our nose, we're also pulling in all those particles. This also makes sense to go ahead and use this as a way of detecting the things that are in that air that we're breathing. Thus, we detect odors in these places. This is also where we're going to modulate our sound, is basically as we are manipulating that air moving through those air passageways. Now, we're going to organize the respiratory system in 2 major chunks. We have or two major ways. We have either structurally or functionally. Structurally, we have the upper respiratory tract, which is going to be things like your nose, your naso, laryngo, and oropharynx, basically up to your trachea, and then below your vocal folds, so from your larynx down, that's going to be your lower respiratory tract. This is why oftentimes you'll hear about people talk about they have an upper respiratory tract infection or they have a lower respiratory tract infection. Different types of viruses and bacteria tend to settle in one of these rather than infecting the entire thing. They'll typically have a preferred region where some of them will kind of stick around up here or some of them will work their way down here. As you could imagine, the lower ones can be a little bit more serious than the upper ones just because we're talking about lungs down here, which means we are in some soft, squishy bits that are very, very thin, so thus could be subject to a little bit more damage. Now, functionally, these upper regions, we're going to call this the conducting zone. Okay, we're going to call this the conducting zone because we are trying to conduct the air towards the site of gas exchange. Our site of gas exchange, our actual respiratory zone is actually going to be down here where it's only going to contain our respiratory bronchioles. Our alveolar ducts is basically a little tiny muscular tube that is going to carry the air down towards the alveoli. The alveoli are these little air sacs down here, these little clusters of grape-looking structures. These are your alveoli. That shape where it looks like these little tiny balls, a sphere has a really, really large proportion of, has a very good proportion of surface area to volume, okay? Which means we can fit a lot of air on the inside, but we also at the same time will have a lot of surface area, which means we can have a lot of room for, or a lot of surface area in order for gas exchange to occur. So oxygens to move from the inside of the alveoli into the blood and for CO2 to move from the blood into that space, okay? So we have a whole lot of surface area thanks to this really heavily convoluted shape. And when I say a whole lot of surface area, I'm talking like the size of a tennis court. Like if you were to take all these alveoli and lay them out in a single layer, it would cover a tennis court. That's a lot, okay? All shoved into this tiny little package in here, okay? So that's what we get by out of having all these little convolutions on the outside of those alveoli, okay? Now, it being connected to a muscular tube is important though, because we don't use all of them all the time, okay? Instead, what we're going to do is we are going to kind of close off these tubes a little bit whenever we don't need to make use of the entire thing. So we can constrict or open these alveolar ducts or these respiratory bronchioles a little bit so that we can regulate how much air is moving into or out of particular groups of alveoli based off of what we need. So say you go to start exercising or something like that, and you're taking bigger, deeper, faster breaths, and you need to move that much more oxygen, all of a sudden, all of these alveolar ducts will open so that you can get air into every single alveolus. This way you can have a whole lot more gas exchange occur and things like that. Now, all this stuff is also open to the outside environment, which means it has to be covered in mucus, okay? We don't trust the things that are outside, so we want to have barriers between the outside environment and the things that are on the inside, okay? So we're going to cover them with a little bit of mucus. Now, this mucus is going to do a couple things. First, it's going to trap stuff as it moves in, so that hopefully we can stop pieces of dust, debris, and things like that from making their way down into the lungs. However, this is also water in a lot of ways, which means whenever we're routing air over this liquidy substance, guess what? We're going to have some liquid move from the mucus into the air. This way we can actually humidify that air as it's on its way down to our lungs, okay? You can actually feel this process occurring a little bit. So if you walk around and say you have a cold, so your nose is stopped up, so you're breathing just through your mouth, what happens to your mouth very, very quickly? It dries out. Exactly. It's drying out because the air that you're pulling in and exhaling is actually just pulling moisture out of all of those tissues in your oral cavity. So your mouth feels really, really dry because that water that should be keeping everything nice and moist is actually just entering into the air that's passing over it and through it. Okay. This is also one of the reasons why it's really efficient for us to breathe through our nose, is because we're going to have special structures in our nose that are going to help humidify that air even better than something like our mouth can, because we're going to get lots and lots of surface area from it. Okay. Now, one fun thing also happens with our respiratory epithelia. That's that they're going to change as we move down. So as we start in the nose, we're going to have pseudostratified columnar epithelia. It will then transition into simple ciliated columnar epithelia, and then finally into simple cuboidal once we get down into the respiratory bronchioles. And then finally, once we are down into the alveoli themselves, they become simple squamous cells. What direction have we just gone? Have we gone from thick to thin or from thin to thick? Thick to thin. Why? Exactly. More protection. We have the thicker tissues closer to the outside environment. Why? Because those are bigger cells and are thus going to protect us a little bit more than these really, really thin cells down here. These simple squamous cells, are you going to get any protection at all from simple squamous cells? No. However, simple squamous cells are super duper thin, which means we can have gas exchange occur rapidly. Okay, because we have that fried egg shape, it's really easy for us to go from outside the egg or outside of it to the other side of it very, very quickly. Just like it doesn't take very much salt to like season an egg, like especially a fried egg. Why? Because they're super, super thin. Like you can wait until you have the egg on the plate to salt it, and the entire egg will taste like salt. Can you do that with steak? No. And it's simply because of the difference in thickness. Okay. That analogy just clicked in my head for some reason. So that's why I said it out loud. It's like, does it sound as stupid to y'all as it does? Like, now, we do actually have a few high abrasion areas, like in our throat and stuff like that, where we're going to have some stratified squamous cells. That's just because we are vibrating things like our vocal cords against one another. So we're causing some friction there. We don't want any of these tissues in places where we're going to have large amounts of friction, just because they would wear away. So these places we will have stratified squamous epithelium. All right, so here we have a picture of those mucous epithelias. So we have our cilia, we have our goblet cells. And once again, these cilia are here so that we can move stuff that gets trapped in that mucus out. Okay. So the example I gave when we talked about cilia was rice. Like I'm really, really bad at eating rice. I wish this was a joke. But it's literally every single time I eat rice, I will inhale at least one piece of rice, probably more, somewhere in the vicinity of my lungs. Which means I will cough and hack every single time I eat rice, because I will have inhaled one of them and I have to get it out. But the way we do that is that piece of rice gets trapped in this mucus layer, and then the cilia on the outsides of these cells start all kind of crowd surfing, and they crowd surf that piece of rice away from your lungs, and they crowd surf up here to your voice box, where then it hits those vocal cords, you elicit a cough reflex, and you cough it out. Okay. And this happens legitimately every time I eat rice, which is unfortunate since we eat rice like twice a week. That's just because rice is awesome. Also, fried rice, super awesome. But it means that I get to do this more often than a real person should, or at least a reasonable person should. You'd think I would have learned by now to not breathe while I'm eating, but I had to compete for food when I was a kid. Not because we were poor, but because my brother could eat more than me. And that was not acceptable. But guess what? I'm bigger than he is now, so ha. He stopped growing at like 5'6". Poor child. He also weighs like $130. It's hilarious. Huh? Yeah, like he was taller than me up until about halfway through high school, and like that's when kind of he pretty much started, like he started maxing out there, and I just kept going. Like I think I was 6 foot tall by the time I was a junior. Like I legitimately grew so fast I have stretch marks on my back. All right, so these are the progressively thinner epithelia. We already talked about that. All right, so now let's go ahead and talk about some of the anatomy here. We've got, of course, our nose. We have left and right side separated by our nasal septum. And then we're going to get into our nasal cavity. Now, your nasal cavity has two major regions. or these big structures right here. These big structures, these big ridges are what we're going to call either turbinates or nasal conchae. Okay, so Conchae, or nasal turbinates. They're called this because if you were to look at them in cross section, so from a frontal section, you would actually see that there's a little bit of a spin to them or a twist. This allows us to take that air that we route in through our nose and cause it to spin as we're moving it through the nasal cavity. So these big ridges give us lots of surface area so that we can get lots of moisture into that air, but also so we have lots of surface area to catch any dirt and debris. My wife's locked out of her car. Awesome. And because the app that she uses to unlock her car is broken and her key is somewhere inside it. Balls. I'll call her in a few minutes while we all are looking at this. Okay. Now, after we get through the nasal cavity, we're then going to progress down into the pharynx. The pharynx is going to contain three regions. We have the naso, oro, and laryngopharynx. The nasopharynx is this top section here, and then we're going to have the oropharynx here, and then we're going to have the laryngopharynx down here. So, nasal region, the short region by the opening of the mouth is going to be our oropharynx, and then finally below the mouth up to our epiglottis right here, we're going to call the laryngopharynx. Here are those nasal conchae. Now we have three of these. We have a superior, middle, and an inferior nasal concha, or the inferior most is the largest, and there's all those kind of these big cavities in between them. Now your nasal or your olfactory epithelium is going to be found up here at the top of the superior one. So as you route the air up into your nose, and that air spins, the top portion of that nasal epithelium is going to be our olfactory epithelium. So it's going to have all of the lock and key style receptors that we have in our sense of olfaction, so our sense of smell. So those are going to be found on that top one. Now we can also see some of the paranasal sinuses here. So we can see our maxillary sinus here. So we can see another sinus here. These are frontal here. These sinuses are going to be very important because as our air is getting routed out while we're talking, some of that air is going to go up into our nasal cavity. And as that air moves up into our nasal cavity, it's going to rattle around in these paranasal sinuses. And as that air is rattling around in those paranasal sinuses, this is what's going to give your voice a sense of tone and a sense of like texture, that sense of resonance. It's going to come from the air being found in these paranasal sinuses. That's also why if you shut off your nose and basically you stop the ability of air to move through your nasal cavity and through those paranasal sinuses, why you lose that resonance. Okay. That's also why people that have bigger heads will also tend to have deeper voices because they have larger resonating chambers. So their voices are going to be deeper and resonate more in their resonating chambers. Okay. So that's what part of why Shaq's voice has such resonance to it. And it's simply because he's got a big ass head. So he's got big paranasal sinuses and thus larger resonating chambers for the sound of his voice. I'm trying to get to a time to where I can do so. So here's a picture of those paranasal sinuses. Here we can see the regions of the pharynx a little better. All right. I'll color after we get through this bit because this is your larynx, aka your voice box. This structure is moderately important. By that I mean it's very important. Okay. The first structure here is our epiglottis. This is a big piece of cartilage here that is going to protect your airway. Okay. This is your trachea down here. So your air is going to get routed through your voice box and then go down through your trachea and make its way down to your lungs. It is covered by your epiglottis. It is covered by your epiglottis because just posterior to your trachea is your esophagus, which is your food tube. That's why it's so easy to have a piece of rice make its way down here. is because it is really just covered by a trash can lid that can move. Okay, your epiglottis is going to basically flop up and down to help us regulate whether or not we have air. If we have air coming in, at which point we can route it through the trachea, or if we have food coming in, at which point we close it. So we just allow it to pass into the esophagus. Okay, that's also why breathing while you are eating and swallowing and chewing can be somewhat dangerous, is because you can try and make this open at the same time that you're trying to send food down to your stomach. Okay. Now, one of the things that we're going to do or to protect our airway is also not just having our epiglottis, but also we're going to hold it open using this little tiny bone right here, which is our hyoid bone. Okay, so we're once again way up here at the top of the throat. Okay, so our hyoid bone is basically going to hold this entire pathway open. It's also important because it's going to be an attachment site for a lot of ligaments and tendons and muscles that are going to allow us to do things like swallow and talk and stuff like that. Okay, now below our hyoid bone is going to be our thyroid cartilage. This is your Adam's, I'm pointing at the wrong screen. This is your thyroid cartilage. That's this big shield right here. Notice though that this big shield is a shield. It is not a tube. Okay, it is open on the back, so it does not close off the back. Instead, it is only going to close off the front. This is also this big shield going to have a big point on it. That point on it is your laryngeal prominence, also known as your Adam's apple. Now, this is going to be larger on Adams than it is E's, but that does not mean that women do not have the Adam's apple. If you feel your throat, you will find a bump at the top of your throat. That bump is your laryngeal prominence on your tracheal cartilage, okay, or on your thyroid cartilage. It's just a little smaller than it is on females than it is on males, okay? Now, the next cartilage down is going to be our cricoid cartilage. So your cricoid cartilage is fun because it's smaller in the front than it is in the back, but it does actually wrap around the entire way, where we have kind of a bigger shield on the back than we do the front. Now, connecting to our thyroid and cricoid cartilages are going to be found a couple of other cartilages on the inside. These are going to be the arytenoid cartilages and the corniculate cartilages on the inside. These are these tiny little kind of buttons in here, and then wrapping around all of these are going to be some arytenoid muscles, okay? The arytenoid muscles in here are going to help us position and move these arytenoid and corniculate cartilages inside the voice box, which means we can use these to change how large the opening is in between these two little cords. If we make it really, really tight together, they're going to vibrate together against one another very, very fast in a high pitch. Or we can pull them apart and they're going to vibrate together in a low pitch. People with bigger throats will have more distance, at which point they can have, it is easier for them to pull them further apart, and thus will speak with a lower timbre, just because the amount of movement that these have are going to vibrate at a lower frequency than will folks with smaller tracheas, at which point they will be closer together, at which point they'll vibrate in a much higher frequency. Oh, one other fun thing, in your tracheal cartilages also do not wrap all the way around. Instead, they're just C-shaped, okay? So they are holding this pathway open, but they are not going to be completely open back here. That's because touching this little pathway right here is also going to be your esophagus. We don't want this spot being hard, because then we would have essentially a hard structure pressing against your esophagus. So if you take a big bolus of food that's trying to move through your esophagus, it would have a harder time moving through it, because it wouldn't be able to stretch this region like it can right now. Like if you've ever taken a big bite of food and not chewed it very well, and you swallow it and it feels like it's tearing its way through your throat, that stretch that you're feeling is in part it pressing against your trachea. Okay, it's not trying to choke you, but it is actually pressing up against your airway a little bit. And that's part of what you're feeling, is it pushing against this kind of empty portion on the back of your trachea. So here we have our vocal ligaments. Here we can see our arytenoid and corniculate cartilages that are going to change how open and closed those are. Now, if you were to actually put in all of the epithelial tissues and stuff like that over these, this is actually what you would see, where so if you had a laryngoscope and you're about to say intubate a patient or something like that, this is what you'd see. If you were intubating a patient, you would want to see this. This is bad because these are not open, at which point this airway is closed. So you can't get any air through here because the vocal cords are touching. However, this is where they are open, at which point you would slide your tube into that hole and pass it down into the trachea, let it sit right there. This way you can get air in and out of here. And also by filling it with a tube, you're going to make it where you can't close this off. So that's what you're actually doing when you are intubating someone. You are literally putting a tube into this little gap right there. Now, once we've done that, you're going to pass down into the trachea. And then the trachea is going to branch into its two major structures, which are going to be your main bronchi. So your bronchi, so here we're going to look at what we call the bronchial tree. We have on the right side, we have a very straight bronchus going into the right side where we have three lobes. The left side, we only have two. So we're going to take that primary bronchus to that main bronchus, and we're going to break it into lobar bronchi, where each lobe of the lung will get its own lobar bronchi. So we have a lobar bronchi here, and a lobar bronchi down here. Then those lobar bronchi will break or split into what we call segmental bronchi. Okay. And then those segmental bronchi will continue breaking until we get into smaller bronchi. That's what's out here. Okay. So we're going to go from main bronchus to lobar bronchi, and then finally segmental bronchi, and then finally into smaller bronchi, and then finally bronchioles, at which point we're getting now close to the alveoli. Okay. Now, the left side is a little different. Notice that the left side has to take a hard left turn into the left lung. Why does it have to take a little bit more extreme path than on the right side? What's right here? The heart. Exactly. So the left side has to deal with the heart a little bit more. So we have a more harsh turn off into the lungs. So we only have two lobes over here. So we only have two lobar bronchi in the left side. Here in the heart, hold on. Which of these do you think is more likely to get the grain of rice all the way down into it? So if I were to have inhaled that piece of rice and then I now have it stuck in my lungs, which one is it more likely to be in? My right or my left? Why? Perfect. That's exactly right. Because this pathway is straighter down, it is more common for things to get lodged in the right lung than in the left, because it's a straighter line. So they will work their way down into the right lung rather than on the left-hand side, because that left side has to take a big turn. Okay. So people will tend to have more serious infections and stuff like that in this right-hand side, just because it's an easier pathway for those things to take. All right, so now we've got down into the bronchial walls. We're going to get down into those bronchioles, at which point we are no longer going to be covered in cartilage, which means we are no longer held open by cartilage. Now we are held open by muscle, okay? Which means we can now control these, okay? We can constrict these to constrict airflow into particular places, or we can dilate them to allow lots of airflow. So we're now getting into the places where we can control it. And this is actually where we get to asthma, okay? Folks with asthma are going to have episodes of what we call bronchoconstriction. That's to say that we're going to go from being open to being closed. Well, if we close these off, that means we're not going to get as much air down into the alveoli, right? Well, that also means that we're going to be running low on oxygen. And when that starts happening, your body is going to start fighting for oxygen. If you're fighting for oxygen while your bronchioles are constricted, this is where you're going to get the wheezing, coughing, shortness of breath, and all that fun stuff that folks that have asthma have to deal with. Oftentimes, this is because of particular airborne agents that can kind of work their way into the lungs, and that's what causes that bronchoconstriction. So because of this, they're going to treat this in a lot of the same ways that they would treat something like an allergy, which is in a lot of cases things like inhaled steroids and stuff like that, which is basically just a way of saying, hey, muscles, deal with this, or even bronchodilators to just help them keep everything nice and open. All right, so once that happens, once we get the air into the bronchioles, we're going to work it down into the alveoli. Once again, these alveoli are going to be these kind of semi-spherical openings where we have this big opening on the inside of the alveolar sac, but this gives us lots and lots and lots and lots and lots and lots and lots of surface area for gas exchange. More surface area means more gas exchange, which means more oxygen in and more CO2 out. So we're going to get more efficient exchange between the outside environment and our blood, both in oxygen and in terms of CO2, which means if you do something like smoke, where you're going to degrade these alveolar sacs, that means you're going to decrease the amount of surface area that you have, which means you're going to make yourself less efficient at getting oxygen into your blood and CO2 out. So those folks will tend to have higher CO2 levels in their blood, and they'll also have lower oxygen levels in their blood, just simply because they're not going to be as efficient at exchanging those things. So exchanging oxygen with the outside environment or exchanging CO2 with the outside environment. Okay. Now, these alveoli are going to, of course, have lots of surface area. We can have that exchange, but that means we also have to have the area for exchange, which remember is going to be the capillaries. So for every alveolus, we have to have a capillary bed covering it to make sure that we can have that site of gas exchange. So we have lots and lots and lots of these. There's something like 300 or 400 million in each lung. That's a lot. That's how we get to like measurements of surface area that equal tennis courts and fun stuff like that, is by having millions and millions and millions of these things. However, they can go away if you do things like smoke or vape or anything like that. Now, in the alveolar walls, we have three major cell types. We have the type 1 cells, which are the most common. These are the squamous cells. This is where we're doing our exchange. We have these type 2 cells. These are fun because these secrete a surfactant. This is going to be important because say I take this piece of paper here, okay? When this paper is dry and I touch the paper together, nothing happens, right? I can pull it apart, put it back together pretty easy, right? Are your lungs dry? No. So let's make the paper not dry. What's going to happen? It's going to stick together. Now, in order to pull them apart, I have to exert effort. And if I were to really soak these things through, I wouldn't be able to do that at all. They're just basically bonded together. Okay? So, do we want that to happen? Do we want this to happen between the layers of tiny, fragile cells in our lungs? Oh, what just happened there? I tried to separate it and it tore. Oh, you'd think that that wasn't actually part of the demonstration, except it is. This is what will happen if your lungs stick together and you try too hard to peel them apart. They can break because it's a single layer of cells. Really, really thin, really fragile cells. So do we want that? Do we want them to stick together? No. Anyone know what this is called when your lungs stick together? It's called a deflated lung. If you've ever heard of someone's lung being deflated, it's because the alveoli had so little air in them and they got dry enough that the tissues stuck. When that happens, you have to figure out how to get enough air into them to make them reinflate. But if you do it too forcefully, like if you just shove a whole bunch of high pressure air in there, it will cause the tissues to tear, just like that paper did, which is bad, that's why whenever that happens to someone, you can't just go and just blow a bunch of air in there and it's fine. It's not a balloon. Okay. So we keep that from happening. We keep these wet tissues from sticking together by doing two things. We keep a little bit of air in there all the time. Okay. You can exhale all you want and there will always be a little bit of air in your lungs. Okay. However, we also are going to cover the insides of the cells with a surfactant. Okay, so we're going to secrete some oil in there. Well, oil and water do not mix, right? So in that wet tissue, so that water covered tissue hits the oil, it bounces off. It doesn't stick because of the oil, okay, from this surfactant. Okay, so this is basically going to coat the insides of these alveoli so that we are going to not collapse. Okay, collapsed lung, that's what I was looking for, not deflated. So that we don't have the tissues stick together should we ever actually get rid of all the air. We're also going to find some macrophages, or what we call dust cells in here, that are going to help find any particular or any microorganisms that work their way in, which, as you can imagine, is going to happen somewhat often. So we've got our macrophages here. Macrophages here, and we've got cells secreting surfactant here. So our alveolar type 2 cells here, and the rest of these are going to be our type 1 cells. Okay. All right. So here's a little bit of information about pneumonia. So what I'm going to do now is let y'all look at some lung physiology and kind of airway physiology, alveoli, and stuff like that with the models on your tables. Okay. We'll do that for a little bit. And then we will go through some kind of breathing exercises and stuff like that and start looking at some volumes. And we'll figure out if any of y'all smoke. So here we have our right and left lungs. So our right lung, once again, is different from our left in that we have 1, 2, 3 lobes. Our left only has 2. And our left lung, we have this little notch. So this is our cardiac notch cut out of our left lung. Now, both of these are going to have their blood vessels and the airway are going to enter in this kind of eye of the bean here. The eye of the bean is going to be called, or be what we call the hilum. Okay, that's where we call this entranceway for pulmonary arteries, pulmonary veins, and airways. So the bronchi. Now, each bronchus is going to have a bunch of segments. So I talked a little bit ago about the segmental bronchi, right? So those segmental bronchi are going to, we're going to have 4 segmental bronchi in the inferior lobe of the right lung, and so on and so on and so on. So each of these colored regions here is going to be the different segments. And then those segments are going to be broken down into individual lobules, and each of those lobules would actually have a different branch of the bronchus leading to those. So we're going to go from a segmental bronchus, which will then break into essentially a lobular bronchus, where it will go to these individual lobules. If you want to look at all of the fun stuff that can happen thanks to smoking and vaping, here's the long list. Increased chance of respiratory infections, so there are genetic changes to the lungs. Emphysema, cancer, lungs, esophagus, stomach, pancreas, stomach ulcers, atherosclerosis, that's say your vessels become harder, which means they don't stretch or recoil as well. So if you have a period of high blood pressure, you can actually just split and tear vessels, which is great. Low birth weight babies and pregnant women, poor delivery of oxygen, nutrients to all systematic tissues, bronchitis, asthma, ear infections. 85% of all lung cancer is caused by smoking. That's great. That's a really big number. Don't smoke, it's bad for you. Don't vape, also bad for you. You could say the same thing for all of these for vaping. People like to think early on that it was safer, like a safer alternative to smoking, just because it didn't smell like smoke. And you could say that it was a little safer just because it's like, oh, it's not actual smoke that's going into your lungs. But 1, the chemical that you're inhaling is probably bad for you. 2, you're still inhaling that chemical into your lungs, which means you can still deal with all of these things. Now, the fun bit is also that those chemicals, as they get routed over those coils, change. Okay. So the whole idea with vaping is that you take the liquid with the chemicals in it, whether it's a flavorant or nicotine or THC or whatever it is, and then you route it around a coil that gets superheated and it causes it to vaporize. And by turning it into a vapor, that you can then have it cross over the boundaries in your alveoli, just like oxygen and CO2 do, so that they're able to get dissolved into your blood very, very quickly. Okay. That's the idea. The problem is that when you route things over a superheated coil, you don't always know what changes you're going to make to that chemical. Sometimes it stays the same, sometimes it doesn't. And it can sometimes break into these weird little versions of chemicals that are way, 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 way worse than something just like nicotine or something like that. And it's just because they get really hot, so that messes with the bonds and all that kind of stuff, fun stuff, so they can do weird stuff. Also, like your water vaping ones are at least going to be a little safer than are the lipid-based vaporizers. People use lipid-based ones because the chemical that they're trying to get is going to be more lipid soluble. THC, so tetrahydrocannabidiol, so the stuff that you would find in weed, that is going to be a lipid. Okay, that's why if you're baking brownies or something like that in it, well, you have to dissolve that stuff in a fat first, so into butter or oil or something like that, rather than just put it directly into the food, because then you won't be able to get as much transfer of it into the food. But the vaporizers are going to be lipid based, which means you're now just not just inhaling water vapor and stuff. Now you're inhaling lipid vapor. So you're inhaling a bunch of fat into your lungs. And as bad as that sounds, that's literally what will happen. You will start to coat the inside lining of all of these alveolar sacs and stuff like that with extra fat, which is real bad. That can inhibit gas exchange and all kinds of fun stuff. This is where you have like high schoolers and stuff like that dying of respiratory diseases and stuff like that is because of more often than not the lipid-based vaporizers rather than the water-based ones. But the water-based ones still aren't healthy. They're still not safe by any stretch of the imagination. They're just not as immediately send you to the hospital dangerous as the lipid ones can be. So don't do that. It's not good for you. If you're looking for those chemicals, there are safer ways to get all of them. Brownies, for instance. Not that I am condoning such things. It is just technically safer. All right. Blood supply. Remember that we have two different types of circulation here. We have our pulmonary circulation, and then we have our bronchial circulation, okay? Pulmonary circulation is going to be where we're getting oxygen into the tissues themselves, okay? Or getting oxygen rather into the pulmonary capillaries. So this is where we're going to be reoxygenating the blood, is in our pulmonary circulation. So this is where we are worried about getting oxygen into the blood. Bronchial circulation is where we are trying to get oxygen to the lungs tissues themselves, okay? Just like the heart, right? How the heart had its own little bit of circulation where we had to make sure that we could feed all the muscles involved in the heart. We have to have some amount of bronchial circulation here to feed all of the lungs tissues, okay? So here we can see our pulmonary circulation. This figure should look pretty familiar since we're going from the heart out into pulmonary circulation, out here, and then back. So make sure you spend some time with this pathway, understanding where the blood is moving, and make sure you kind of understand what's happening to the blood once it's getting out here into the lungs so that we understand this portion of the pathway. We're also going to have lymph nodes and fun stuff like that in here. Now, just like we had with the heart, we're going to have a couple pleural membranes. So we're going to have a visceral pleura and then a parietal pleura. Remember that the parietal pleura is going to be the one on the outside, and the visceral pleura is going to be the one that's actually touching the surface of the lungs. And that we're then going to have a little bit of a space in between them. It's very important that these do not touch, or that they do not grind against one another. So we are going to have some lubricant in here to keep that from happening. Getting air in that cavity is bad. This is actually what is called a pneumothorax. Okay, so if you've heard that on Grey's Anatomy or whatever, that's basically what has happened. Or what has happened here is that we've gotten air in that space in between the pleural cavity, or in between the lung and the parietal pleura. So we have air in the space when we shouldn't. So that can lead to some problems. Collapsed lungs, we've already talked about here, and that's where basically we have some problems getting the alveoli torn apart. Mm-hmm. Yes. Yeah, it's extra fluid. There's always going to be some, but it's more like the difference between having some condensation on the outside of your water bottle versus having the water in the water bottle, right? The fluid in the lungs is basically like we're looking at the water rather than what is always there, which is basically going to be that little bit of condensation that we need. Does that make sense? That's kind of our differences in volume that we're concerned about here. Now, once again, in order for all this stuff to work, the air outside your body is going to have to be different than the air inside your body. Okay, so the atmosphere, the atmosphere is going to contain oxygen, so more oxygen than will, say, your blood, at which point we're going to move that oxygen from that air into your blood. So we're looking at this red line here. Okay, so the red line is oxygen. The blue line is your CO2. So because the air outside contains more oxygen than the inside air does, that will pull oxygen into the blood. Okay, As the blood moves around to your various tissues, it will drop off that oxygen into those tissues. At the same time that it's dropping oxygen off to those tissues, it's going to pick up, or the air in, or sorry, your blood will be low in CO2. Okay, because the blood is low in CO2, and it's low in CO2 because it just left the lungs, that's going to cause the higher levels of CO2 in those tissues to suck some of that CO2 into the blood. The blood will then carry that CO2 back to the lungs, and then, well, the outside environment doesn't have as much CO2 as your blood does, so it will pull the CO2 out of your blood. And we're going to continue doing this over and over and over again. Do not think, although this, I actually don't like this figure because it seems like that's what it's saying, is that we are exchanging CO2 for oxygen. These two things are happening separately. Okay, if I put you in an environment that had the same amount of CO2 as your blood, the oxygen part would still happen. These two things are completely independent. It just so happens that we have them traveling on the same highway. That's all. Okay, they are two different cars that just so happen to be utilizing the same road. Okay, air outside has higher oxygen concentrations than your blood. Because of that, we pull oxygen into the blood. That blood then moves out to the tissues, at which point it drops off the oxygen. As that blood returns to your lungs, it is now low in oxygen, right? Since we just dropped off the oxygen in our tissues. So now that we have a low concentration of oxygen in the blood, what's going to happen when it comes into contact with air with a high concentration of oxygen? We'll do it this way. I'm going to draw it in two circles because it makes more sense. Okay. See, outside has high O2. Inside. Do your tissues have a high concentration of oxygen or a low concentration of oxygen in comparison to the outside environment? Look. So we have a low concentration of oxygen. Okay. So what happens here in the lungs? The blood comes in. So blood comes in, finds this. So this blood is currently low on oxygen since it's coming from the inside of our body, right? So we've dropped off oxygen to the tissues. So that means we're going to be low in oxygen. So we have a low concentration of O2 as we get up here. So what's going to happen? O2 will move into the blood, right? So now this blood has a high concentration of oxygen, right? So now it will carry it to the tissues. High versus low. So what direction will the oxygen move? So oxygen will go out into the tissues. So from the blood to the tissues, right? So now our blood, is our blood low or high in oxygen now? Low. So now we have low oxygen level blood returning to the lungs, where it comes into contact with the outside environment, which has a high concentration of oxygen, which means oxygen gets pulled from the outside environment into the blood, carries it back to the tissues, drops that oxygen off, returns, grabs more oxygen, returns to the tissue, drop it off, returns, grabs more oxygen, returns to the tissue, drops it off, and so on and so on and so on. Okay? This is what is happening with the oxygen. The CO2 is basically doing the same thing in parallel because the outside environment here has a low amount of CO2. Okay, so this blood with a low level of CO2 goes down to the tissues where we are doing aerobic metabolism, right? We're taking glucose, which is C6H12O6, and then converting it into H2O and CO2. That is our equation, right? You make CO2 as a byproduct of breaking down sugars and fats. You make ATP, you are going to produce CO2. Okay? So that means if we are doing that in our tissues, which we are going to produce CO2. So we're going to have a high concentration of CO2. right? Now that we have a high concentration of CO2, where does that CO2 going to go? Into the blood, right? So now our blood will have a high level of CO2 and it goes back to the lungs. Well, now what happens to that CO2? It will go out to the outside environment. So now this will be low CO2, right? So now blood that is low in CO2 will work its way down to the tissues where we have a high level of CO2 and it will pull it into the blood, go back up here. Well, this is higher than this, and now that's the direction that the CO2 is going to go. It's going to go out into the outside environment, right? This is what is happening. We have two different pathways. We are not exchanging oxygen for CO2. Okay? If you get rid of this, that will still happen. If you get rid of that, this is still happening. These are completely independent. Okay? The reason they are independent is because they are relying on two different pathways of getting to the things. Okay? We use oxygen, yes, to help us do this process, but oxygen is not involved in this particular change. Okay? When you put the oxygen here, so to help us do this, this oxygen goes here, not into the CO2. Okay. The reason that's important is because these gases are going to act independently. So we have to treat them a little bit independently. All right. Now, when it comes to breathing, different muscles are going to be involved in different types of breathing. So ordinary quiet breathing, so just your normal, like y'all just sitting in here, you're doing normal quiet breathing. So you're breathing in, breathing out. This is all happening with your diaphragm, which is, there it is, that muscle right here at the base of your rib cage. This pulls down, changes that volume. That's what's going to allow us to pull the air in. And we're also going to have a little bit of external intercostals pulling your rib cage out just a little bit. Okay. That is your ordinary quiet breathing. Diaphragm relaxes, external intercostals relax, closes the rib cage back off just a little bit, just because it's no longer pulling itself open. That makes the volume smaller, so that's going to allow us to exhale. Okay, no muscles are actually contracting in a normal quiet exhale. Okay, now that normal quiet breathing is usually moving around 500 milliliters of air. Okay, so it's usually moving around 500 milliliters of air. So that's you have a water bottle. Actually, about this. No, hold on, that's 32 ounces, so it's half of that. So that's to about here from the bottom. Okay, so an ordinary like 50-cent water bottle from Walmart is going to be your half liter. This is 1 liter. Okay, that's how much air we're normally moving just on a normal quiet breath, is that half a liter. That's all it really needs. Okay, now I want everybody to take a big, deep breath. Pull in as much air as you possibly can. Notice that you have other muscles moving. What are you doing? You're pulling up on your rib cage. Your sternocleidomastoids pulling up. We have our scalene muscles, which are back here, pulling up, trying to elevate your rib cage. Why? Because you're making your rib cage even larger. Your external intercostals are really trying to pull your ribs out, so much so that they're getting help from your pectoralis minor and your erector spinae. So we're pulling the rib cage up and out in the front, and we're pulling the rib cage back in the back. So we're really trying to stretch the rib cage out as much as we possibly can. That is forced inhalation. We can make this do something like 3.5 liters, which is 3 of these. And that is in someone that is not trained. A trained athlete can get 6. Which is stupid. Well, it's awesome, is really what it is. Like, it's just a massive volume of air that these people can move. And it's simply because we're using all of these muscles, just truly opening these areas up as much as we possibly can. Okay. Now, I want you to breathe out as hard as you possibly can. Not just as fast, but push out as much air. So just exhale and keep exhaling and keep exhaling and keep exhaling. So your body kind of twists, your body kind of bends forward a little bit. Why? Because now your abdominal muscles are going to be working. Your transversus abdominis is pushing in on your gut to help push your diaphragm further up. Your external obliques are going to help pull everything in. Once again, trying to put as much pressure on your diaphragm as you can to push it higher up into the space. Your internal intercostals will also start kind of constricting to pull everything in. So fun, fun, fun. All of this is to change the volume of the thoracic cavity. Okay. For ordinary quiet breathing, we're looking for 500 milliliters. We're only looking for this much air. We don't have to move very far. You don't have to try. Okay. Diaphragm moves down and it can move down this far pretty easy on its own. External intercostals move that far just a little bit, make things a little easier. We're looking for small volume changes. That's all we need in order to get enough oxygen in to keep all of our tissues doing everything that they need ordinarily. Okay. So by making it a little bit larger, well, that means we're going to have a smaller volume, a larger volume, less pressure, and the pressures are going to try and equalize. Okay. So that's what's going to pull air in. When we make the volume smaller, well, volume smaller means we're going to have higher pressure, and well, higher pressure is going to then cause that air to move out. And we're going to do this over and over and over again. Larger movements will cause larger moving of air. Okay, so if you need to move more air than that quiet breath, you just simply need to make the difference in volume even larger. So you have to open the volume more. And that's where we get those big things. Okay, this idea of increased volume leading to decreased pressure or decreased volume leading to increased pressure, this is what's known as Boyle's law. Okay, this is the relationship between volume and pressure. That's this P1V1 equals P2V2. This has to be the same. So if we increase the volume, well, if those things have to be the same, if we increase the volume, that means we have to decrease the pressure, or vice versa. If we increase the pressure, we're going to have to decrease the volume. Does that make sense? In order for the two things to be equal. If those two things are not equal, what changes is the movement of the air. Okay? Because Boyle's law is really meant to be in this kind of vacuum, like where the air cannot escape. Okay? Our air can. It can escape because we have an open tube connected to it, which means we also have to deal with pressure gradients, where we have air in one place and air in another. When we decrease this pressure here, the air wants it to be the same. The air is looking for equilibrium, right? So if the air is looking for equilibrium, well, if we've decreased this pressure here, that's going to pull air from out here to down here until those two volumes are equal, right? Or until those two pressures are equal, which means when you take that breath in, you will pull in enough air into your lungs to make the pressure inside your lungs equal to the pressure outside every time. That's why it takes a moment for you to breathe in. It's just because you're pulling these particles in until the pressures equalize. Okay. Now, does anyone see a problem? Anyone know what altitude we're at right now? We're at a few hundred feet above sea level. That's it. Not very far. Huh? Yep, exactly. But that also means that the pressure that's outside your head's a little different, which means when you take that breath in, you're not going to pull as much air in as you do down here because there's less stuff in it. There's less pressure, which means there are less particles of oxygen and CO2 and nitrogen in the air at altitude, which means is your quiet breathing of moving half a liter going to be enough, or changing your volume by half a liter? Is that going to get you enough particles of oxygen to do what your body needs to do? No. Which means you can't just do quiet breathing anymore. What do you have to do? You have to breathe harder. You have to start doing deep breathing just to sit still, which is fun, unless you have virtually no metabolic requirements because you're really just kind of sitting and vegging out and stuff like that. But if you say go into even higher areas, or you increase your physical activity and you need even more oxygen, you have to pull in even more air, which means you have to breathe in even harder. Okay. But that's because as we get into altitude, we have thinner air, we have less pressure. Sea level is kind of where we are right now. We have 760 millimeters mercury. Everything works fine. Once you get up there, you actually get a lot less. Because the changes in pressure that we're looking at are not very high. In normal quiet breathing, we're only decreasing our pressure by 4 millimeters mercury to get to our half-liter change. 4 out of 760. It's not very much. Okay. In order to generate this amount of difference, when we have less pressure outside, means we have to try a little bit harder. Now, all of this is, of course, going to be controlled in our medulla oblongata because it controls things like breathing rate, respiratory rhythm, and all that fun stuff because they're also looking at oxygen and CO2 in places like our aorta and our carotids with those baroreceptors and chemoreceptors and things. It is monitoring all this stuff so that we can decide if we need to breathe quietly or if we need to breathe deeply and all of that fun stuff. Dude, I have no idea. Yawning, I still don't understand. I need to look into it a little bit more because I think there actually has been some research in the last few years, like learning a little bit more about it, but it is something I still don't quite understand. I've heard that, but I've also heard that that wasn't linked together, so I don't actually know. I will do some reading on it because I haven't looked into that stuff for a while. I'll do some reading on it and I'll get back to you on Wednesday because we're still going to be talking about respiration and stuff on Wednesday. If you've heard of apnea or sleep apnea, this is basically temporary cessation of breathing during sleep. So you might have fleshy bits or something like that cutting off airways, or just breathing may stop momentarily, at which point you'll tend to see people that don't sleep very well and they'll kind of shudder themselves awake because they stop breathing basically and they realize they stop breathing. So they'll startle themselves and like take a big deep breath in to try and get that whole process going again. You tend to see this in folks that are overweight, have high blood pressure and fun stuff like that, or folks whose names are Matt and Mosley and all that fun stuff. Folks that snore, this tends to be a problem. But one of the things we're monitoring here is going to be the partial pressures of CO2. Okay. When CO2 levels get high, that means we need to exhale more. Okay? When CO2 levels get low, we don't have to worry or spend as much time exhaling. Now, one of the ways that we monitor this is by actually monitoring the pH of the blood. Okay. Because CO2 in the blood is going to be moving around as carbonic acid. Okay. So it's going to make the blood more acidic. So we can monitor the pressure, the amount of CO2 in the blood by monitoring the pH of the blood. When the pH gets low, that means our CO2 levels are getting high, which means we need to exhale more. The opposite of this is also true. When CO2 levels get low, that means the pH of the blood is actually going to get quite high. When that happens, you will need to exhale less or rebreathe some air so that you can bring those pH levels back down. And this is actually what happens when people start hyperventilating. So when people are hyperventilating, they're breathing super fast, they're breathing super fast, they're breathing super fast, they're breathing super fast. So they're pushing out a whole bunch of CO2. So their CO2 levels in their blood get low, which means their pH levels start to get high. So their blood starts to get alkalotic. Okay. That's also why you can take that person who is hyperventilating and give them a bag to breathe into. And when they start breathing into that bag, what they'll do is they're going to start rebreathing a bunch of that CO2, which means they're going to pull additional CO2 into their blood. And after a few breaths, they'll have gotten enough CO2 into their blood that their pH levels will be back down to normal. So into that 7.35 kind of range or 7.34 range where they were getting close to that 7.4.5. So they'll bring those levels back down and all of a sudden you'll notice that their breathing gets slower and things like that. It's because we've actually gotten us back to a normal level of CO2 in the blood. That's why breathing into a bag helps when you're hyperventilating. Simply because you're rebreathing that CO2 and you're bringing the acid-base levels back into balance because you're getting enough CO2 back into the blood. You're usually not hyperventilating because you need more oxygen. It's usually because of a stress or something like that. It doesn't have anything to do with oxygen. That's why a few breaths into a bag isn't going to kind of have you shut down because you're running out of air or anything like that. When you haven't gotten rid of enough CO2, that's when you're going to do things like sigh. So like big, deep exhales are going to help you push out a lot of excess CO2. Breathing really fast and like rapid exhaling and stuff like that will also help you get rid of some CO2. But one of the things that will happen with kind of like a sigh reflex, it's like after kind of a stressful moment, like as part of that stress, you've increased metabolic activity in your body. So you've actually produced a little bit of excess CO2. So your CO2 levels will get high and you'll have that sigh movement, that big exhale, but so that you can actually get rid of additional CO2. So you can bring those levels back up. Mm-hmm. Yep. Yeah, isn't it great? Yeah, fun stuff. When we watched the new Inside Out movie, it was like, this is moderately traumatic. So one of the things that we can actually call this hypoxic drive. Okay. So levels of oxygen in your blood can be a stimulus for breathing. Okay. So when oxygen levels get low, so your oxygen levels in your blood get low, that can be a stimulus that we need to breathe more deeply. Which means like you're going to notice that you're going to start getting low whenever you exercise like quickly. Like, so we're going to store some oxygen in our muscles, especially our fast twitch muscles, so that they have access to oxygen now, so that they can do strong movements very quickly, very powerfully, and things like that. But after a few moments, they will have burned through that, and you now have to start getting oxygen to those tissues. So your blood starts to get low on oxygen, at which point, here in a moment, I'll have to start breathing harder and deeper to bring the oxygen back into those muscles because I've exhausted their myoglobin or the oxygen stored in their myoglobin. So we bring those oxygens down. So I now am somewhat hypoxic. So I have to breathe more so that I can bring those oxygen levels back up in those muscles. Does that make sense? I've gone into oxygen debt in those muscles. So I have to replenish that oxygen that I've gotten rid of. So there's some fun stuff in here talking about sneezes and coughs and fun stuff like that. I like this chapter. This chapter's actually got some cool stuff. Flow rate is also going to depend on the differences in pressure. Okay, so if we need to move more air, we can move more air by increasing the amount of pressure gradient or by decreasing resistance. So how could we decrease resistance in the lungs? Are all of our tubes in the lungs completely open all the time? No. Some of those are made out of muscle, right? So if we want to decrease resistance, because we're on the bottom of the math equation, right? So we are pressure difference. So delta is difference or gradient. P is your pressure. And then R is on the bottom. So because it's on the bottom of your equation, if you make this number go up, you make this number over here, your flow rate go down. So if the bottom gets bigger, your flow rate goes down. So if you increase resistance, flow rate will go down. If you decrease resistance by opening your bronchioles, well, so if we open our bronchioles, that makes a bigger tube. That means there's less resistance to the air moving through it, right? Bigger pathway. Well, that means we're going to increase our flow rate. So say we need to increase the amount of oxygen that we're getting in a breath, we take a deeper breath, a bigger breath. So we make a bigger pressure differential because we're opening the cavity up larger. And we open up those bronchial tubes by opening up those smooth muscle cavities, opening those up, decreasing resistance. That means we can get more flow. That's it. Okay. If we increase the pressure gradient, we generate more flow. If we decrease resistance, we get more flow. If we do both, we get even more flow. So say we need a whole bunch of flow, like in the case of exercise, what are we going to do? We're going to breathe harder And we're going to open up more of those alveoli so that we can get more air moving through them. So we're going to decrease our resistance at the same time. Does that make sense? This is also why folks that have asthma, exercise is tough. Because guess what they're really not good at doing? Decreasing resistance. Their bronchioles are going to constrict. They're going to reach some threshold. They're going to breathe in a piece of dust or something like that, and bronchioles will constrict, at which point this goes up. Resistance goes up a lot, which means flow rate goes down. Even if you do this part as hard as you can, because of the resistance here, the flow rate will still be low, and they will start coughing and hacking and all that kind of stuff and have to stop the physical activity because they're simply not getting enough air into the tissues. Part of why asthma can suck. I've had multiple friends that had asthma. I had one friend actually have an asthma attack because he caught a giant fish one time. Like he got so excited like afterwards, or like while he was fighting the fish, he like started coughing and hacking and stuff like that. Like started having a minor asthma attack like while he was fighting this like 8-pound plus bass. It was great. He was fine. Like we got the fish in the boat and like he took an inhaler. But it's like we're looking at this fish and he's damn it, Kyle. Good fish, buddy. Also knocked off like a 12-pounder one day off of his line. He was pissed. I'm not very good with the net, as we learned that day. Also, here's the thing. If you're going to do something, like if you want to be able to do something like in a high pressure scenario, practice it first. The first time I had ever picked up a net to try and net this fish, it was like a 12-pounder. So I, of course, knocked it off because I'd never done it before. I didn't know what to do. Yeah, it was great. All right. Now, so we talked about resistance. Your lungs also have what we call compliance. This is basically the ability of the chest wall and the lungs that can, like, their ability to expand. Okay, their ability to expand and then kind of relax. Okay, that is compliance. The easier the lung expands, so the stretchier it is, the greater the compliance. So if your lungs are nice and stretchy, they're going to have more compliance, which means whenever you breathe in, they will follow that breathing in better because they'll stretch more and they'll be able to get faster ventilation into them. Now, that normal small breath, like I said before, is called our tidal volume. Okay, so our volume that we're breathing in is our amount of air per breath, so that is our volume. Our respiration rate, so how many breaths we're taking. If we want to figure out how much ventilation we're getting, all we're going to do then is take that volume of air that we're moving per breath, multiply that by the breaths per minute, and that's going to give us a rate of flow per minute. Ordinarily, this is going to be around 6 liters. Okay, so half a liter, we typically breathe around 12 breaths a minute. This brings us to our typical rate of around 6 liters a minute. This can go up or down based off of your own personal size and all that kind of fun stuff. But it's usually going to be around the 6 liters, and this is what it takes in order for us to get enough oxygen to our tissues. Okay, and that is at sea level. Okay, once again, that is at sea level. Now, we're going to have a little bit of what we call dead space in here. So there's around 150 milliliters of dead space, so portion of the lungs, at which point we have no exchange of respiratory gases happening. Okay, the rest of that stuff is going to be happening down here in the alveoli. Okay, so in the alveoli, we're going to do around 4.5 liters of exchange every minute. If we breathe deeply, we can then get a larger proportion of ventilation here. We're going to measure this using a spirometer, and that's actually what we're going to do now. Now, the 4 volumes that we're going to be looking at here are inspiratory capacity, functional reserve capacity, vital capacity, and total lung capacity. Total lung capacity tells you, well, what it says. This is the sum of all of the volumes in your lungs, including this thing that we call residual volume. Residual volume is here, and this is a number that we cannot change. You are going to leave some amount of air in your lungs all the time. This is going to help make sure that we do not collapse our lungs, okay, that our alveoli do not touch themselves. So we have to keep a little bit of air in there just to make sure that the pathways are open enough that we can get more air in. Okay, so that's what we're going to call our residual volume. Okay. All right. I want everybody to take the biggest breath in that you possibly can. pull as much air in as you can. That is your inspiratory capacity. This is your normal breath, which is your tidal volume, and then your inspiratory reserve volume. This is a measure of compliance. This is basically saying how stretchy are your lungs. If you smoke, this number will not be very big because this is going to make everything nice and firm, and they're going to limit then how much air you can push into those cavities. Okay. Now, when you exhale as much as you possibly can, you're pushing out as much air as you possibly can. That's going to be what we call this expiratory reserve volume. This means how much air are we keeping in the lungs all the time after a normal quiet breath. Okay. Now, if we take a normal breath, which is our tidal volume, and then our inspiratory capacity, so we're going to basically take our 500 and then we're going to add our inspiratory capacity above that. And then we take our expiratory reserve volume and add all those together. This is going to get us to our vital capacity. Okay, that's basically to say how much air can you exchange through forced breathing, whether through forced inhalation and through forced exhalation. This is going to be one of the big numbers that we're going to measure today. That's because it's actually easier to measure this than it is individually do inspiratory capacity and force exhalations and stuff like that. Okay. So what we're going to do, this is also going to allow us to kind of calculate our forced expiratory volume. This is basically to say, after normal breath out, push out the rest of your air. That pushing out the rest of your air is going to help us distinguish folks that have chronic obstructive pulmonary disorder. Okay, so COPD, things like emphysema, because those folks are going to find it difficult to expire. Folks that have restrictive things like pulmonary fibrosis, where their lungs have gotten hard, these folks are going to find it difficult to inspire. Okay, smoking can actually lead to both of these. Another reason why it's bad to smoke. Have I hammered that point enough that it's bad to smoke? I hope so. Good job, France, for making it still popular. All right, so what we're going to do then is we're going to look at some of these numbers. All right, so to do this, we've got our barometers back here. Okay. So this is just a little doodad. All right, all we're going to do is take the small end of that and push it onto the small end here. And then I want you to take the little dial and you're going to turn it to where that little needle is pointing at the 1,000. Okay. First thing we're going to try and measure is our tidal volume. So what I want you to do is I want you to take a few, just like normal breath. Okay, and at the end of one of those normal breaths in, I want you to then exhale just a normal amount of air out. Okay. This should be pretty darn close to that 500 line. So it should have moved up to pretty close to 1,500. So halfway in between the 1,000 and 2,000. Now, this is hard to do because it's hard to breathe out through here at a normal pace. So if it's a little above or a little low, don't think too much about it. It's probably going to be around 500 milliliters. Okay. So that is your tidal volume. That is your TV. So go ahead and write that down. So whatever number you got, in my case, it was literally 5. Okay. So that is this. That means on your normal breath in and breath out, and you can do a few and kind of take an average. That's the smarter way to do it. Do a few, take an average. So just normal breath in, normal breath out, breathe out, then do a few more, not through the thing, and then do another breath out through the spirometer. So this is literally measuring volumes of air. So cc's of air, cubic centimeters. Now, after you've done that a few times, I then want you to breathe in as much as you possibly can. Yeah, now I want you to turn the needle down where it's facing at the 0. And I want you to just breathe in as much as you possibly can. And now I want you to breathe out through here as much as you possibly can. This is this volume right here. What we just measured as vital capacity. Your force inhale all the way to force exhale is your vital capacity. This is your VC, so vital capacity. Mine was 6,000 milliliters. Like on the button. I had a football player in here, I guess a year or so ago now, and they got this thing to spin around. Like literally back to the start and past it. It's like 7.5 liters. Like, good Lord. Where is it? Oh, did it go back to the 0?
Oh wait, no, mine went to 63.
Okay.
I just reset it.
So like 3,800?
Yeah, 3,900.
Once again, though, this is a measure kind of a volume of air that we're moving. If you are a smaller person, this volume will be smaller, okay? Just simply because you do not have the space. And if you want to do this a few times and once again get an average, that's totally fine. But now remember that your vital capacity is a combination of your inspiratory reserve volume and your expiratory reserve volume. So now what we need to do is we want to measure either your inspiratory capacity or your expiratory reserve volume. The easiest way to do this is going to be to do your expiratory reserve volume, okay? So now what I want you to do is just take a few breaths, like back to normal, just do normal quiet breathing. And then on one of those normal quiet breaths, like after you breathe in, I want you to exhale and then just keep exhaling until you can't exhale anymore, okay? So just on one of those breaths in, push out all your air. And that's going to get to your tidal volume and your expiratory reserve volume. So what we just did is tidal volume plus expiratory reserve volume. And in my case, this was 1,500. Okay. Now, like I said, this also included a normal breath in, right? So now what we need to do with this number that we just got, we need to get rid of the tidal volume. We have it right here, right? So what do? Subtract, just maths, okay? So to get rid of the tidal volume, we're just gonna subtract our TV, which means we're going to subtract 500. And that's going to get us to our expiratory reserve volume of, in this case, 1 liter. Okay. So 1,000 milliliters, 1,000 cc's. Okay. Well, now we can calculate back calculate from our vital capacity our inspiratory reserve volume. So inspiratory reserve volume is going to be our vital capacity, so 6,000, minus our tidal volume, minus our expiratory reserve volume, which is this one here, right? So in this case, so my ERV, and all of this should equal our RV, so 6,000 minus 1,500, so that means I have an inspiratory reserve volume of 4,500 milliliters. Once again, I'm probably bigger than you, so don't think too much about it. Like, size matters here. Vital capacity minus total volume minus expiratory reserve volume. And that's how we're going to get to our inspiratory reserve volume. So your inspiratory capacity is basically your total ability to inspire. So that's going to be your tidal volume plus this big number up here. What we just calculated here is this number in yellow. So this kind of region up here, that's what we just calculated. But your inspiratory capacity, so how much can you breathe in, is going to be that number plus your tidal volume. And it says a normal value in females is around 2,400 milliliters, and males it tends to be around 3,600. So there is a chart right here in your book. So this is figure 23.25. I can also just blow this up if y'all wanted to look at your numbers and see how they compared. They say comparison is the death of joy, and that's true until you realize that you're better. But I'm also an elitist. ERV stands for expiratory reserve volume. So that's this one right here. So once again, now y'all can see the normal values over here. Now, like I said, smokers, vapors, and things like that can also see these numbers be quite different, where you'll see some of these things get a little smaller. Folks with restrictive disorders are, once again, the folks that are going to have a problem inspiring. So pulmonary fibrosis, so fibers in the lungs, make it where your lungs are not going to be as pliable, which means they're not going to be as stretchy. Because they're not as stretchy, these folks are going to have a hard time getting air in. So those inspiratory reserve volumes are going to be lower. Okay, for folks with things like pulmonary fibrosis. Folks with obstructive diseases are going to have a rough time getting air out. Okay, yeah, things like emphysema. So that means they're going to have a problem getting air out. So they're going to have less compliance. Okay, so they're going to have less ability to get that air out. So they're going to have a smaller expiratory reserve volume, to the point where a lot of times, like, you'll see them breathe air in. So like they'll be able to breathe air, they'll breathe in 500 milliliters, but they won't be able to exhale 500 milliliters. Some of that air just gets trapped in their lungs, and you trap and trap and trap and trap and trap. And then you have a big coughing, hacking attack and stuff like that to try to push that stuff out. That pushes that out, that extra air finally, and then they'll do this over and over again. So that's why they tend to have these bouts of repeated big coughing events and stuff like that. It's because essentially they're filling their lungs up with more air and they can't get it out. So they have a big cough until they get some of that air out, and then they breathe back in. And every breath they trap a little bit more air, and then they cough some out. And then they do this process over and over again. It's just like essentially walk themselves up and then crash, walk themselves up and then crash. So sounds super, super fun, I'm sure. But that is like literally what's happening there, and why that can be such a problem for those folks. That's also why folks that have things like your emphysema and stuff like that tend to have this real barrel chested look to them a lot of times. And that's simply because their lungs are just always hyperinflated. All right. I think I'm going to leave it there for today. And when we come back next time, we will talk more about what is actually happening in the individual gas exchanges. Anybody have any questions on anything? I want you to hold on to these numbers. Actually, no, we'll look at different numbers when we come back next time. We don't need these. I was going to say, like, I can't really compare these to whenever we have y'all exercise. But next time I'll kind of have y'all come in. We'll do some more walking exercise, and we'll look at respiratory rate and stuff like that. These things can just go in the trash. They're fine.
Not bio.
Not the biohazard. It's fine.